Hi everyone, my name is Pedro Gonzalez-Ochoa, and today I'll be talking about my applied project titled Engineering a Biosynthetic Pathway for Sustainable Monoethanolamine Production in E. coli. Let's dive into the problem statement. Did you know that in 2021, industrial process plants in the US alone generated 165 million metric tons of carbon dioxide gas that went straight into the atmosphere? Here's 1 metric ton of carbon dioxide gas for your reference. It's a lot. That's where our target molecule, monoethanolamine slash MEA, comes into play. It functions by trapping CO2 gas in a chemical reaction, which is pretty helpful, but it is highly energy intensive to produce it and has its own CO2 emissions. Here's an overview of what our MEA production currently looks like. As you can see, you have ethylene oxide that you feed through a bunch of different chambers and control the temperature, flash it, pressure, and then you get monoethanolamine, diethanolamine, triethanolamine. So it's a long, strenuous procedure, but it is currently how we do it. So here is a overview of what my project is hoping to accomplish. We get some CO2 runoff from refineries. We run it through this chamber that's going to have a whole bunch of monoethanolamine that's been synthetically produced through this metabolically engineered E. coli, and then it's collected and harvested and disposed of, or taken away and to use in whatever form. And how we're going to do that, you might ask? Here's the metabolic pathway for MEA from glucose in E. coli. As you can see at the top, you start off with glucose, and through a series of different enzymes shown in blue arrows, you convert into intermediates that ultimately get you to MEA. The genes of interest that we're going to highlight in this experiment is SDAA and UBC, which you could see on the right side. We delete those, and we modify the SERA gene to get us to the precursor molecule L-serine. And then we're also going to be playing around with the serine decarboxylase at the bottom, SDC, turning serine into monoethanolamine. Here's our experimental design for the plasmids that we're going to be chemically transforming into E. coli. You can see the ones that we use in the experiment. Going more in depth into the plasmids that I just mentioned, you have 15 right here, which is taken from common spinach you can find in the kitchen. Thale-cress is 16, and then we had a codon optimized version, which you can see right here on the left. This is a plasmid map using Benchling, a software that we use to map out all the important genes. So this is the overview for how chemical transformation works. You get your plasmid DNA, mix it with these competent E. coli cells, and then after letting them mix, you heat shock them, and then that allows the plasmid DNA to enter the membrane, as you can see down there below. And then the E. coli cell will transcribe, translate all of the sequences, and produce whatever enzymes we require it to. So in this case, STC, SERA. Here are some results from my experiments. You could see that after testing the spinach codon optimized, as well as the Thale-cress SDCs, we had two pretty competitive strains. We had the codon optimized spinach as well as the Thale-cress, which directly correlates with MEA production with its serine degradation. So it directly converted this substrate into MEA, and they also grew the best. So after taking time points every 24 hours for 4 days, this is what our trend looked like overall. And although I would say that those are some pretty promising results, we did have some setbacks. We noticed that there was a missense mutation in one of our strains. So that means that one of the nucleotides that previously coded for an amino acid changed. So this mutated from, a T to an A or a C to a G, and it affected the production. So it wasn't what we originally had planned. So we're going to have to, unfortunately, redo the experiments, but shouldn't be too hard. And that concludes my presentation. Thank you for taking the time. I'd like to give a big shout out to my PI, Dave Nielsen, Cody Kamoku, my undergrads, Eric and Francesca, for all their help, as well as the neighboring Wong Environment Lab. Thank you.